Tissue culture is the growth of tissues or cells separate from the organism. This process were done in a laboratory environment that is typically facilitated via the use of a liquid, semi-solid, or solid growth medium such as broth or agar by in vitro methods and under sterile conditions. Tissue culture, also known as micropropagation, where the tissue culture process helps to grow multiple uniform plants in quick succession. Tissue culture of banana propagation has gained popularity because of its several advantages over conventional methods. Tissue culture is the best method for propagating bananas. It ensures they are true to type and free from pests and diseases. Banana is typically propagated vegetatively. Thus, tissue culture as a propagation technique provides a robust means to prepare disease-free planting materials that can provide the first line of defense in developing an integrated disease management program for banana. Different techniques are established to propagate bananas in a lab environment. These include the shoot and meristem culture, callus culture, cell suspension, and many more.
In tissue culture propagation of banana plantlets, we first start with collection of suckers. Next is disinfect the suckers before performing inoculation on culture media. After inoculation, the suckers are allowed to proliferate. Brooding and hardening are then carried out before field planting. So let's go to the first step, which is collection of suckers. Different stages of banana keikis about 40 to 100 cm tall that are free of banana bunchy top virus symptoms can be collected for tissue culture. The presence of virus can be studied in the laboratory. Banana suckers selected are removed to obtain approximately 4 inches or 10 cm of inner pseudostem tissue containing meristem. Meristem consists of undifferentiated cells that can develop into all the other tissues and organs that occur in plants. But what is pseudostem? The pseudostem is the part of banana plant that looks like a trunk. It is formed by the tightly packed overlapping leaf sheath. The second step is disinfection of propagule. First, wash the pseudostem with running water to remove soil and surface tissues such as roots and sheaths. Immerse the pseudostem in a container of undiluted household bleach, which is 5.25% sodium hypochlorite for 30 to 45 minutes. They are then surface sterilized for 10 minutes each in 4% sodium hypochlorite followed by 0.1% mercury chloride, with 3 rinses in sterile water in between each treatment. Decant the bleach solution and keep the surface sterilized pseudostem in the container. The third step is inoculation. Murashige and scoop medium is used in this technique. The ingredients for tissue culture media of banana shoots growth include MS macronutrients, MS micronutrients, MS vitamins, FE-EDTA1, BAPS plant hormone for growth, sugar, coconut water, and agar. During the inoculation step, the MS medium is prepared with the ingredients previously mentioned. Medium scalpels, forceps, cutting plates, and magenta boxes are autoclaved according to standard autoclaving procedure. Perform all procedures under surface sterilized laminar flow hood. The pH of medium is adjusted to 5.8 with 1.0 molar natrium hydroxide or hydrochloric acid prior to autoclaving at 121 Celsius for 20 minutes at 100 kPa. The surface sterilized shoot tips are trimmed and placed on full-strength Murashige scoop medium. A study in 2015 has found a cost-effective media for large-scale propagation of banana. For carbon sources, they discovered that table sugar 3% resulted in greening earlier than rock candy and small candy. Meanwhile, for water sources, early greening for RO water is only 5.4 days. Greening for borewell water is 7.8 days, while greening for single distilled water is 7.0 days. The fourth step is maintenance of shoot cultures. Observe the cultures for contamination. Discard contaminated cultures as soon as contamination is noted. Observe for browning and bulging of corm tissue. Weaning of leaf tissues and growth of shoots during the first month of culture. When shoots coming out are almost 2 cm tall, the shoot tips are ready for subculture. Keep shoot cultures in air-conditioned room under a 16-hour photo period.
Step 5 is proliferation. The shoot is transferred into fresh medium culture when propagule is 2 cm in size. But if the shoot is overgrown, it will be less proliferative. However, if the shoots are more than 2 cm, we can make a longitudinal cut at the tip of the growing shoot. Next, in a sterile condition, subcultured onto MS medium with a supplemented BAP 6-benzyl aminopurine to prevent nubbins production. This is because the medium does not contain auxin. The number of proliferated shoot recorded after 3 to 4 weeks of obtaining sufficient shoot during subculture process. The subculture is repeated, less than 5 cycles. If the cycles is conducted more than 5 times, it can lead to mutation such as dwarfism or elongation. Step 6. Rooting. Table 2 shows the banana rooting medium. The ingredients of rooting medium is prepared based on the MESCO 2005. The ingredients are MS macronutrients, MS micronutrients, MS vitamins, ferric EDTA which consists of ferric ion and EDTA, sugar, coconut water which is optional, and agar. Next, the small plantlet will be obtained after the last cycle of the subculture process. The root needs to be separated and transferred to rooting medium. And lastly, it is ready to be planted in the soil after 3 to 4 weeks. The plantlet will start to have roots as well as the leaves. Step 7. Hardening. Hardening is a process of preparation for plants before transplanting. Plants need to adapt in a new environment such as changes in the amount of light, temperature, and altitude. The plantlets have shoots and roots, but the shoots and roots are not able to support themselves when we transplant them in the soil. Therefore, the vessel's lid need to be removed and exposed to partial sunlight at least 10 days for in vitro hardening process. Plant tissue culture is fragile due to growing under low intensity, artificial light, and high humidity. Next, after the process of hardening, it will be transferred to the greenhouse. Plant tissue culture must be washed to eliminate the agar medium that's stuck onto it, because the agar has sucrose and other nutrients that can be used as a substrate for disease-causing bacteria to develop. Firstly, we need a potting mix with good moisture holding. Potting mix is a sterile and safe for plants because there is no pathogen. Second, we need to fertilize with liquid fertilizer. Third, the banana plant is placed in a partially shaded area for 2 weeks before fully expose it to the sunlight. Lastly, the banana plant needs to be placed in a condition where there is no aphid present. Aphid is an insect as shown in the figure. The last step is planting. Planting is a process after 2 months of acclimatization process in greenhouse with the height of plant which is 8 inches. A bit dry soil is better because it helps the roots to attach more to the soil when we start transferring the plant from its container. A sucker might produce after a few weeks due to excess of growth regulator. Every 5 days, a leaf will produce during early development. The plants are ready to be harvested within 9 to 10 months after planting in the field.
So, what we can came up with the discussion is that micropropagation of banana will produce high number of uniform and pest-free banana plantlets, which are beneficial to the farmers and also increases banana exportation to other countries, which also increase the country income as a whole. An example of a country that profit from banana exportation is India, as it is one of the largest banana producers in the world. Tissue culture propagation of banana plant must be done meticulously to ensure its optimum quality and zero contamination. Next, we are going to compare tissue culture method versus conventional sucker method to grow banana. Banana tissue culture method will produce disease-free and clean plantlets, and it also has high rates of multiplication. It has rapid and uniform plant growth and shorter harvest-to-harvest period. It will produce banana of higher bunch weight, and the yield production will increase annually. Other than that, it also only requires small space to multiply the plant. Meanwhile, banana sucker method requires less input. It is not as fragile as tissue culture plant where it needs extra care and special condition. Banana suckers does not need it. It also can be immediately planted on ground. Both methods has their own perks. Banana tissue culture method is highly suitable to be applied by large banana producing company, while banana sucker method also can be applied for home planting.
Then we're going to see the advantages and disadvantages of this plant tissue culture. On the advantages of this plant tissue culture, it produces matured plants quickly. Second, it reduces the chances of transmitted diseases and pathogens. Produce exact copies of plants with desirable traits, and mass propagate plants for commercial use. On the advantages of this plant tissue culture, it is complex and expensive process and require sterile conditions for regenerations. The procedures are depends on the type of species cultured and more chances to contaminate without proper care. Plant tissue culture offers a number of advantages over traditional pig culture, including independence from geographical, seasonal, and environmental variations, and also short growth cycles.
Next is the critical factor influencing in vitro growth. First is the choice of explant. In many species, explants of various organs vary in their rates of growth and regeneration, while some do not grow at all. The choice of explant material also determines if the plantlets developed via tissue culture are haploid or diploid. Also, the risk of microbial contamination is increased with inappropriate explant. Thus, it is very important that an appropriate choice of explant be made prior to tissue culture. The most commonly used tissue explants are the meristemic end of the plant, like the stem tip, auxiliary bud tip, and root tip, because these tissues have high rates of cell division and either concentrate or produce required growth regulating substances, including auxin and cytokinins. Some explants like the root tip are hard to isolate and are contaminated with soil microflora that become problematic during the tissue culture process. Certain soil microflora can form tight association with the root system or even grow within the root. Soil particles bound to roots are difficult to remove without injury to the roots, thus allow for microbial attack. This associated microflora will generally overgrow the tissue culture medium before there is significant growth of plant tissue. An alternative for obtaining uncontaminated explant is to take explant from seedlings, which are aseptically grown from surface sterilized seed. The hard surface of the seed is less permeable to penetration of harsh surface sterilizing agents such as hydrochloride, so the acceptable condition of sterilization used for seeds can be much more harsh than for vegetative tissue. Next is the explant size and thin section culture system. In an earlier study, Rosliman have shown that the production of ORCID protocol in vitro can be substantially improved by manipulating the size of the explant alone. For example, the number of protocol produced by thin transverse section, which is 0.6 mm thick, derived from a single shoot tip, which is 6 to 7 mm long, was 5 times greater than that produced by an intact shoot tip, which is 6 to 7 mm long culture under identical condition. A similar observation was also made recently in sugarcane. In this crop, leaf explant produced numerous plants, which are more than 50 per explant, when the thickness of the explant was reduced to 1 to 2 mm. This finding clearly indicates that the explant size plays a key role in the expression of organogenic potential of the culture tissue. This explant size-based difference in organogenic capacity has since been successfully utilized to develop thin section culture system, a novel approach in plant regeneration. The last one is in vitro environmental condition. Light quality is one of the potential important environmental factors, as it has been shown to affect the direction of plant morphogenesis in vitro, and it also plays role in between gametophytic and sporophytic pathway. Tissue culture processes often involve extensive cutting and stress injury of tissues, which may cause physiological changes in plants affecting the success plant tissue culture.
From this experiment, we can conclude that when compared with the conventional approach, tissue-cultured bananas will yield higher growth and net income. Besides that, a somatic embryo is a strong tool for producing high-quality planting materials in large quantities. Lastly, plant yield and vitality in floriculture species can be increased.